So this video is a quick recap on tissue culturing. It's part of that chapter on cell diversity. And in this short video, we'll just go over what is it and what do I need to know for my exam? Are there any particular details I need to know? Start off by writing a definition. Tissue culturing can be defined in a number of ways. So let's define tissue culturing. It's the growth of cells in or on a sterile nutrient medium. Or you could write, it's the growth of cells in vitro. That means in glassware or outside of the body. So there are older definitions that were previously accepted. For example, the growth of cells outside an organism or the growth of cells outside of the body were previously accepted in 2019. Best to be current. You must also give two applications of tissue culturing. Let's start with a plant application first, and this is micropropagation of plants, producing many plants from a small piece of plant tissue. So a piece of plant tissue is removed from the parent plant and transferred onto or into sterile nutrient medium. After a while, a callus forms. A callus is a group of undifferentiated cells, and this means that these cells, this callus, has the potential to grow into any type of plant cell. So the callus is treated with growth regulators, types of plant hormones, and eventually a plantlet is formed. And in this way, from a small piece of plant tissue, you get many identical plants. Mostly somatic cells, so not gametes, normal body cells are used, and in this way you produce many identical plants from a small tissue sample very quickly. When you combine tissue culturing with genetic engineering, there are infinite possibilities. Imagine producing all of these plants, identical plants with the same special traits. Essential conditions would include sterile conditions, nutrients, growth regulators, a suitable temperature for plants, light, and those gases, carbon dioxide and oxygen. You must be able to give an animal application. You could give IVF, in vitro fertilization, or you could state the production of skin for skin grafts for those patients who are burns victims or those patients who have leg ulcers. So in growing animal cells, they require oxygen and they also require a temperature of 37 degrees Celsius, body temperature.